Welcome to the final talk of this year's ISMB ECCB in Dublin, which I felt was a fantastic meeting, and we're going to have a fantastic talk to end it. Imagine you were in a world, you looked at a sequence database, and the sequence database were full of errors, and you had new knowledge that you wanted to add to that sequence database. And you tried to influence the people who do this database to do that. And they're not responsive, absolutely not responsive. And then you talk to your supervisor. Supervisor says, let them do it, don't do anything yourself, and then you start a database. That, actually, you may not know that, but that started the word annotation. The root of the word annotation is actually Amos PC. In some sense, that's the Greek spelling of this. Amos started this database that annotates proteins, this database that in some sense started it all, SwissProt. Then came a moment when suddenly there was this bad decision, no funding for SwissProt. So that was the unique thing that had changed the world, and suddenly there was no more funding for a unique thing. And then Amos invented something new. The procedure kill the fax machine. So you invite the world to support you, and this is, I believe today, like as if you would call for people to submit all their genomes on hard disks to a certain place. Thousands of hard disks coming at the same time, that would also be an overload. That clearly is a very big thing and makes you famous. It's a big breakthrough. But this is still a small community. Now imagine that they make a toy that is supposed to look like you. That's the real breakthrough. And Amos achieved that one too. He achieved many things. He started many things. And on top of all of that, he is full of energy to do new things. And this will give you a great keynote talk to end the meeting. Amos Bairoch.
So, I want first to start by thanking, Burkhard for this really wonderful, introduction. But I also want to thank the ISCB for three things. Giving me the honor of being a fellow of the society last year, and unfortunately I could not be there last year to receive the fellowship plaque, but I got it later by mail, to invite me to this meeting. But I would say the third important thing is to allow me to hear the talk, which should, I think, have been the keynote talk of the end of the meeting. This wonderful talk, and I know a lot of you were there, from David Searles, with the parallel between James Joyce, Earlis, and bioinformatics. I'm blown up with, by this talk, and it's an hard act to follow. So I feel a little bit, donkey starting this talk, having heard this wonderful talk by David. So sorry, this will be less, entertaining than that talk for those which were there. I will try to describe what we do now in the CALIPHO group. So the CALIPHO group was founded in 2009, and this was just after we had completed in SwissProt the first round of annotation of all what were, at the time, the list of human protein. Why is this the first round? Because annotation never ends. That's, in fact, one of the good and bad thing of annotations. The good thing, because there's always going to be work to be done. You always want to put more things in knowledge. You want to encapsulate the knowledge which is progressing. The bad thing is that it's very hard to fund because it's a never-ending process. And funding agencies don't like this type of things. Anyway, 2009, we started this new group with two missions. One is to develop a resource called neXtProt, which I will describe in a few minutes, and I will describe new development of this resource, but also to do something else, which is to carry out laboratory experiments on a selected set of protein, of uncharacterized human protein, to discover their function. Now, why was that? Because when, at the end of 2008, when this first annotation run of the human proteome took place, what was obvious, and this is from a slide from a meeting in Siena in September 2008, that we were confronted by the fact that how little was known on the function of human protein. Of course, our protein, you could write books on it, as complex books as Ulysse from James Joyce. You could write a book on insulin, on P53, but there are those where your book will be very shallow. And as a joke, I put up this, pie chart, which is basically the status of knowledge of the protein at that time. And a lot of it was, of course, prediction, expectation, and a lot of wishful thinking on what some protein should do. Now, of course, it's not as bad as this, but still there was a lot of protein with no function. This is still true because it takes a lot of work to go through a function. And I will maybe spend 2 slides on the effort we do in the lab, which have nothing to do with bioinformatics, except they're assisted by bioinformatics. And just to maybe do this in contrast with the bioinformatics world, which is the bread and butter of this meeting. So we have a pipeline which is very classical. We use a number of different very classical techniques, PCR, cloning, IPs, yeast to hybrid, GST pull down, overexpression. At 3 points, we use bioinformatics to take all of the clues we can get from what is available in high-throughput experiment, co-expression, phylogenetic profile, 3D structure prediction, everything we can take our hand-ons, and we're very keen to try every method of function prediction. Generally, this gives us a few small clues, and then we go on and have experiments being done in the lab. Now, in contrast to what we hear a lot during this meeting and preceding meetings all over the world, this is not big data, it's not high throughput, it's tiny knowledge. And just in one slide to recapitulate, to show you how slowly it takes to do this, we have 4 lab stories completed, meaning that we have 4 papers which were published late 2012, 2 in 2014, and 1 early this year. We have 2 stories which are not yet completed and which are still being worked on. And so basically, what can we show in 5 years? More or less 5 proteins characterized. And I wouldn't say they're fully characterized. We just have more clue and more information on them than when we started. Now, that's, of course, obvious for all of you which have worked in labs. It's less obvious for a lot of people which come from a computing world, which go into bioinformatics, and think that those characterization efforts can be very fast. All of them were works of PhD students, postdoc, which took 2 to 3 years to go through. The only thing we do differently from other people doing this in other labs is we study protein nobody's interested in. So we took boring protein, which had no association to disease, no association to any pathways known, protein which nobody could know what they did because they had no recognizable domain or they were not very interesting and so on. And so this is only a tiny bit of effort in this big, drive to understand what those human proteins do. And don't worry, there's still a lot to be done. We keep track of all the protein where we think people should characterize, and there's still 2,000 of the 20,000 human protein where there should be a lot of effort being done to uncover their function. So still 10% of the human protein, we don't know anything about what they really do. Okay, so let's go now to neXtProt. So neXtProt, we wanted it to be a type of one-stop shop for human protein so that people could visualize on the web server the information we had on those human protein, but also to extract what they needed and to export the relevant annotations so that they could use it in their own tools, and also to perform complex and precise queries. And I will go back to this because, to be honest, in the first 4 years of neXtProt, we were not really good at allowing people to export annotation, and we were really bad at allowing them to do complex and precise query. But this has changed now. So just to explain what we do with neXtProt, we start with UniProt KB, SwissProt, all of the human protein, which continuously get updated and re-annotated. We take the release of neXtProt, and then we populate neXtProt with different type of information coming from different resources, from Ensemble, from Cosmic, from BG, from a lot of control vocabulary and ontologies, and a lot of information also on proteomics. I will come back to that in a few minutes. And basically, this construct neXtProt. So on a busy slide that you will see here, where I put some statistics of the release. Currently, we have 20,000 human protein, which is what there is in UniProt in terms of human protein entries. Of course, this translates to more sequence because of splice variant, alternative promoter, alternative initiation. So we take all of the SwissProt annotation, and then we add a lot of different information. I won't have time to discuss all of them. I don't want to give a talk really on all of what there is. We do a lot in terms of proteomics. I will come back to that. We do a lot in terms of getting variants into neXtProt, but this is only the beginning. So we load data from dbSNP, from Cosmic, and we take a lot of information from a wonderful resource, which is the Human Protein Atlas from Matthias Uhlen, which has not only expression data through antibodies for the protein, but it has now a lot of other information on substrate location. Basically, the Human Protein Atlas keeps growing, not only in terms of protein, which are covered by it. Now there are, I think, 16,000 to 17,000 of 20,000 protein, but also in terms of all what Matthias and his enormous team in Sweden are providing to communities. So we owe a lot to what Matthias Uhlen is doing to build, neXtProt. Now, okay, so you have all of this data. What also is important is we do a quality filtering at we put, in fact, 3 different bins, gold, silver, and bronze. And our expectation is that what we think is gold should be with an error rate of less than 1%. What is silver is between 1 to 5. And what is bronze, we don't want to have inside neXtProt, it's more than 5%. So in neXtProt, you see only 2 of those 3 tiers, the gold and the silver. And you can toggle back and forth and look at annotation which are either gold or silver. And you could say, how do you do this? Do you have statistics and so on? In some cases, we do have statistics. For proteomics, you can use FDR, you can use different type of statistics. For some other methodology, what we do is it's a dynamic process and we work with the data provider. For example, with HPA, we sit with the people from HPA, we look at all the grades, antibodies, quality, what they decide to do, and so on, and it's an iterative process. In some case, for example, with the people in BG in Lausanne, which provide a meta-analysis of the data which is in Array Express, we went through the different level of confidence they had, and we decided together of what could be the 1% mark, where should we put the limit between gold and silver and between silver and bronze. So it's imprecise. This 1 and 5% limit is what we would like it to be, but so it's basically something which is dynamic process, and we go back and reanalyze data depending on what the provider or other the users tell us about it. Now, I told you I wanted to speak about proteomics, but that would be just 3 quick slides. You maybe have heard of the Human Proteome Project, which is called HPP, Human Proteome Project, through the UPO. It's a UPO initiative, and it has 2 components, one which is chromosome specific and one which is biology and disease specific. Now, it sounds a little bit weird to have a chromosome specific project for protein, because once the protein, of course it's encoded in a chromosome, but once it's in a cell, it doesn't care really from which chromosome it comes from. And doing it from a disease perspective, from a tissues perspective would make sense, but from a chromosome, it doesn't really translate into a very scientific, idea. But it's, I think, a good political idea, and you need politics in science. Basically, allowed UPO to farm out the work to different countries, each of them adopting 1 or more chromosome. So see the chromosome project more as a type of placeholder to get people to try to do things at UPO, and then go back to their country by saying, I've done the work on my chromosome, and can I get more money to do this type of proteomic work? So it is chromosome-oriented, but in a way, it's not really scientifically oriented towards the chromosome, but politically oriented towards the chromosome. An interesting concept, I would say. Now, in the context of HPP, what we do is we basically, as we have been selected by UPO to be the knowledge resource of the HPP project, so we integrate results which are tagged to be from HPP. Anybody can participate in HPP and says that a set of human proteomic work is targeted toward HPP, and say put it into the Proteome Exchange system through PRIDE and Peptide Atlas or GPMDB. And then we basically take this information. We, in fact, use Peptide Atlas as a resource for where the peptide, identified peptide comes in, because they provide us with a recomputed and reanalyzed set of all the human peptide. And we provide metrics to tell people to which level, we are now in terms of having seen all of the protein that are supposed to exist in the human body. Now, I won't have a lot of time to discuss this, so I will just show one slide with a proteomic view of neXtProt with different tracks. And we put data on the antibodies, which are, which part of the protein sequence is used to make antibody by Matthias Uhlen, what he called a protein ESTs, the peptide, which are identified, through different studies, and the SRM peptide, which are used to monitor protein abundance as a type of tools to proxy to what is done at RNA level of RNA-seq, which is more and more used by proteomic studies to monitor protein concentration. Okay, so let's leave the proteomic view, and you can go yourself to neXtProt and look at the different views, the expression view, the disease view, the sequence view. Now, I was telling you, we didn't really have something to search really neXtProt in a nice way. So we built a new search engine, which currently is on a separate server than the main server, but it will be, as we were building all of neXtProt, then it will migrate to the nextprot.org server once we finish rebuilding also the web portal. So it is a search engine with two components, a simple Google-like search, I will just have one slide on it, and a more advanced search capability, which is based on a SPARQL RDF technology. We also have a new API, which allows you to retrieve any type of information we have in neXtProt up to the level of the experimental condition in XML or JSON. So that allows also people to build application on top of neXtProt. We'll also have a new version of the XML export format. I'm saying it's in progress because we basically still need to round up some bugs, and so XML currently it's complete, but it's not finalized or new XML, and it will be finalized probably end of August, beginning of September. But it's already available on this search.nextprot.org. And later we want to also have an RDF version of neXtProt. So just, if I go on the simple search, nothing fancy, you type in words or lists of words, and you get, you can search, of course, in the protein in neXtProt, but also in terminology, because we integrate a lot of terminology, going from Go terms to a lot of other terminology resource, and I will come back to one terminology resource that we're providing now, which is quite unique. But you can search also in a publication, and then you decide if you want to search in the gold-only data or gold and silver. So basically, you want to include the 5%, the data which is between 1 to 5% error rate. You get either a summary, a list of protein, or your more detailed one. You can filter by different things, which protein have a 3D structure and expression profile, and so on. You can save this as a list. I will come back to it, and also sort your result by different criteria. Nothing fancy here. I guess all of you have done, which are developers, would say, yeah, well, no problems there. And I agree, that's not the big part of what we were rebuilding. The big part is we wanted to do advanced search. So what do we mean advanced search? I thought the best way was to give you a few examples. For example, I would like to have all the protein which are located in mitochondria with an experimental evidence showing that it's originating from the Human Protein Atlas, or from DKFZ using GFP, but not from other methodology. Okay, that's, complex, but not so complex if you have access to the experimental details and so on. Now, here is one which I find more complex, because you need to go to a graph to do this. You want all protein with a PDZ domain, that's easy, but that interact with at least one protein which is expressed in the brain. So you're doing a query not only on the proteins, but on the interactor of that protein and what they do. Or here, protein which have one or more negatively charged residue inside a transmembrane domain. So we can do queries on any sequence pattern inside a domain or a region. You can do queries with numbers. I want a protein with 2 transmembrane followed by a SH3 domain, which has a proline inside, but no cysteine, and so on. So basically, we started with a list of questions we wanted ourselves to have to ask neXtProt, a huge list of questions, And the software developers said, well, we have one solution, use a SPARQL endpoint, and they did this. So here is other examples. I mean, oops, what did I do? No. Let me get back. Yeah. Okay, sorry. So next one, protein from 2 to 4 transmembrane. Or here, we can use the data we have on the genetic, loci of the protein and their place on the genome. You want protein whose gene is less than 50,000 base pair away from the gene coding for P53. So we can do queries of this type. And I will show you another query, which is important because, in fact, it also opens up what we're trying to do now, which is we can't integrate everything, but we can do federated queries. So this last query is quite important. We want all the protein which are target of antipsychotic drug and highly expressed in brain. We have the information of which protein is highly expressed in brain, but we don't have the information that they're target of antipsychotic drug. But we can do a federated query with drug bank. So basically, from the SPARQL endpoint, we can do this federated query to whoever has a SPARQL endpoints and share the same accession numbers. And as we use the same accession numbers, systems and UniProt, most of resource which tie up with protein have links to UniProt. So we can do queries with drug bank, with PDB, with Campbell, to give a number of examples that we have used already here. Now, people can just basically write either as a SPARQL query, get their answers, and so on. But we know that for a lot of you which are developers, SPARQL queries are easy to do. But for the biologist, forget it. It's going to be, and myself, I consider myself more a bioannotator and biologist than a bioinformatician. And yes, I can now write SPARQL queries, but it was a steep learning curve. And I know that most people won't do it. So what we thought is if we populate the system with enough examples, people can then modify those examples because it's easier to modify something that already exists and to build your query. So we already have 120, soon 130 queries, either that we made or that users have made, because we ask users if they're willing to share their queries. And then people can then go on those queries, edit them, save them under their space, and either provide it to us so that we can add it to the collection, or if they don't know how to do the queries, they can ask us for help, and we'll do the query for them and ask them if they're willing that we put this query in the query library. And so that's a nice thing. And then, of course, we need to have with this a list management tool, which allows you to either take any of your queries and save them as a list, but take any of the lists that you have yourself built through any type of transcriptomic, genomic, proteomic experiment, and allows you to manage those lists by mixing them, by doing OR, and not. Nothing very fancy here, but it's quite useful because the queries and lists for us are synonymous. And of course, you can then save your protein completely in XML or in JSON. You can only save part of it and so on. Now, I know a lot of you are computer developers, are software developers. So what we did is completely rewrite, as I said, the neXtProt backend. We used to have a very complex system. Now we do everything to an API service, which is sitting on Spring. We have the database, which still is Postgres SQL, but we have this SPARQL Advanced Search in Virtuoso. We use a full text search with Solar, nothing special here. And we basically write the website, on top of the API. Now, and of course, you can build your custom client application on that. The thing which is also different is that all of our code is open access and available in GitHub. So basically, we don't keep any software development directory ourselves. We put everything on GitHub. Search for neXtProt, and you will have different components of neXtProt and the GitHubs for that. And our software developers are really eager for people to use it. We also are starting to write viewers because we're going to rewrite the website, and we have viewers on top of the data. And as we're writing them, they're not even available now on our old website, but you already can get access to them, and you can use them. Feel free to use them for other things. So we have viewers for peptide, for people who are doing proteomics. We have a viewer which we call a feature viewer, which is being built by a student, which is using the API to build this viewer. And this will show any type of features on a sequence from PTM, variant, and so on. And still in level of, for developers, we have a snorkel front end, and you can basically play around with all of the I mean, it's a SPARQL endpoint with the snorkel front end. And as I said, please don't hesitate to contact us. In every page, it says contact us. And all software developers are really eager to see people start to use the API and the tools. So it's really an open, we want this to be an open community system and not to build ourselves basically all of the tools that can be built on top of neXtProt. Now, on another subject, so I'm leaving now the part of which is totally neXtProt, we build another tool, and I will explain why. We build a tool to help us to annotate, which is called the BioEditor. It's a web-based biocuration platform. It's not something really wonderful in terms of software architecture. In fact, our software developers tell us that we should rebuild it from scratch because it's quite ugly in terms of code, but it does what we need to do. We wanted to be able to do annotation in a highly structured manner, and basically using triplets or extended triplets. Now, with a data model which is quite flexible, and we can basically quickly change to represent multiple data types, and I will give you examples. And also, we wanted to be able to really capture everything in terms of the source of annotation, meaning not only, of course, the PubMed, paper or DOI of whatever was the source of the annotation, but everything in terms of experimental criteria. Without over-engineering it, because of course you can end up by having so much metadata, as it was said, I don't remember which, I think it was Philip Roca, which says that in one talk that, you end up, or it was maybe ending. Anyway, somebody said that, rightly, that if you have too much metadata and you're asking to get all of this metadata, you end up having nothing. So it's a compromise. And we can export those annotation either in XML, but this system could be tailored to produce RDF or open bell statements. So each annotation consists of a triplet with a subject, relation, and object, and it can be extended with what we call precision. Our subjects are what we call bioobjects. They can be a protein isoform, a protein variant, a protein complex, or a protein group. So if you want to say that actin does something and you don't know which actin, because there is a lot of different actin in the human proteome, you can make a protein group, define which protein belongs to this group, and tie up an annotation to a protein group. And of course, you can do it to a complex, to, as I said, an isoform, a variant, or a PTM. And then we have a number of relations, which we have developed in an ad hoc manner while we were using this, and I will tell you why, how we're using this system. And then you have an object, which can be itself a bioobject, so another protein, another variant, and so on, or a chemical, a KBI, a gene ontology term, a human phenotype ontology, a disease, and so on. And we can have precision, like, for example, lean phosphorylate BTK producing BTK page here 551. So we have this system, and then we have, and this allows you to do annotation like here, BRCA1, low 63 decrease binding to EPD1, knowing that this is not how it's stored. Everything is stored as accession numbers to the relationship, to the bioobject, and so on. So annotators see text, but behind it, there is no text whatsoever. It's all, controlled vocabulary and control identifiers. So we have annotation, and we have evidence, from one to many evidence for the annotation, and of course, the evidence use eco code to the type of experiment which were done. It says eco plus because we in fact have provided a lot of eco terms to eco, which have put them into a subsection of eco saying SIB. It's not SIB specific. You can use those eco code, but we wanted, we needed a lot of eco code from the group, so they created this section. We have biological models which tell you where the experiment was done. Is it a cell line? Is it, a tissue? What species it was done on? The protein origin, because it can be different. You can work in a human cell line expressing a mouse protein, and so on. Of course, the reference and things like that. And basically, this is stored in this, this BioEditor allows us to capture this type of things. Now, what did we use this BioEditor for? For two projects. One, which is a one-off, and this was a project which you haven't seen yet the result, but we're nearing the time where we can provide it to you. Why don't you see it yet? Because it was a project done with industry. We did it in collaboration with Merck Serono, which asked us to do a full human proteome, a full human genome annotation to go deep in all of the human protein kinase and annotate them for their drug screening platform. Unfortunately, you may know that, but in Geneva, we had a big, section of Merck Serono was in Geneva. They closed down completely the Geneva site, which means the project was interrupted, but we still had funding which allowed us to do 300 of the 500 protein kinase and to do 30,000 annotation abstracted from about 14,000 papers. So it's a big corpus, and what we had upfront negotiated with Serono is that we could provide it to the community after an embargo, and this embargo will finish in 2016. So that's why I'm talking about that, because in a few months we'll be able to liberate this huge corpus of data on protein kinase. But the other one, and which is part of the title I gave to this talk where I spoke about SPARQL and variome, is we wanted to do a lot of protein variation annotation. Now, we're not annotating which variant is implicated in which disease, because there's a lot of effort being done and so many talks were targeted towards this subject here. But what we're doing is we're currently doing deep annotation on phenotypic effects of protein variation. We're doing this with two projects which we had funding. One was to do this on a number of genetic cancer, BRCA1, BRCA2, and the Lynch syndrome, another one on sodium channel. So the selection was more based on which funding we got. But the idea is we're using this as a type of prototype of the effort we want to do. So we capture really deeply phenotypic annotation of the effect of variant from literature. We don't invent anything here. It's nothing special, nothing predicted, but we wanted to make sure that people have a training set of very highly curated phenotypic effect of variation on protein, which are involved in disease, and it can be genetic disease, or soon we'll do some of it for cancer. Now, so that was the goal, so to help improve clinical interpretation by providing to people developing prediction tools and to people using already this corpus, gold standard phenotypic annotation. And this will appear in neXtProt at the end of the year. We needed a system to basically put it in the new website. As our new website is not yet ready and our new database layer is still being worked, it's frustrating, so it's taking longer than I expected. It's like everything. You always want things to be faster. But I hope that in November or December, we'll be able to push those annotations to the community and go along, and of course you'll be able to download them in XML, do whatever you want with them, no string attached. Just to explain, to give an example, one slide on example, for BRCA1, a lot of the annotation have to do with, of course, the function of BRCA1 or the association of BRCA1 with different proteins. So here you see a number of annotations. There were, for example, 388 variants annotated with more than 1,000 phenotypic annotations. So a lot of variants have more than one phenotypic effect. And we also annotate if the phenotype is not modified. So for us, it's as important to say that something has been modified by variants, and also to capture that something has not changed with this variant, in a specific position. So we also annotate normal phenotypics, so saying that with this change in sequence, nothing has changed for one of the phenotypes that we are, in fact, capturing. So you see, depending on the different things that BRCA1 does, we have a number of, of annotation which covers a different aspect of BRCA1. And this will be true for all the different type of protein which we'll annotate. We started with BRCA1 as a proof of concept, and also because there's a lot of data around it. Now we'll spend do I still have 5 minutes, or? Yeah. I will spend 5 minutes on a pet project of myself. As Burkhard was saying, I wanted to clean up protein database, still interested in cleaning up protein database, but when we were doing neXtProt, we wanted to have a resource on cell line. And to be honest, I knew a little bit about cell line, but I didn't know that it was such a big mess. Now, this led to the development of something called the Cellosaurus. And to tell you, with the world of cell line, of course, you know that cell line are used all over the world by labs. There are a huge number of cell collection, ATCC, DSMZ, Recon, and so on, ECCSC. But most people, and we're guilty also in our lab of doing this, you go to the next guy and ask you, can you give me this cell line? And you trade cell line and so on. So a number of them, people buy them from the cell collection. A number are traded between labs, are distributed by labs, and so on. Now, there is a number of variety of cell line. I just put that for those which know less about cell line, like I was also not knowledgeable of the different cell line which I use in labs, so I don't feel bad re-explaining to people which are as naive as me. So, I mean, of course, cancer cell line, transform cell line, embryonic stem cell, hybridoma, and so on. There are cell line, I mean, catalogs from those resources like ATCC and so on, but they concentrate on cell lines they distribute, of course. They're not going to advertise cell lines that they don't distribute. There are ontology, like the cell line ontology, CLO, or, for example, branded tissue ontology has a lot of cell line. And there is also a number of other EFO, for example, the experimental factor ontology has also cell line. But each of them cover a number of cell line, are sometimes redundant. For example, CLO has many entries for the same cell line. For each place where the cell line is distributed, they have one entry for it. And there are many specialized databases for cell line. But we couldn't find a single resource where we could find all the cell line we needed to describe in our annotation project. So that led to Cellosaurus. Oh yeah, another thing which is important, sorry, I forgot about it. It's a huge mess in terms of names, standardization, origin, and so on. And all you know about all of this, I mean, horrible story which happened in 1970s and 80s with ELA contamination, which contaminated so many cell lines, but it's still going on. And it's a sociological problem. You have people which are working on cell line for 20 years. It's been 19 of those 20 years that those cell lines are known to be contaminated. They still publish saying that their cell line come from prostate when you know that it's come from a melanoma, to give one example. Now, this, I mean, is recognized by the community working on cell line, but we wanted to put this also in it. So we built a Cellosaurus, which is a thesaurus of cell line, with all of the immortal cell line we could find on it, and also natural immortal cell line like stem cell, iPSC, or finite cell line when they are distributed and used widely, like through Coriell and other things. Its scope are vertebrate cell line, with emphasis, of course, on human mouse and rat, but also now insect and tick cell line. It doesn't include primary cell or plant cell line. Just current depth of information, so we provide, of course, a category, which type of cell line it is, the sex of the individual from which it's derived, which is not easy to get, and it's important to know if you're working on a female or male cell line, what it expresses as genes, its expression profile will be different, and a lot of people don't give you this information. Species of origin, the breed subspecies, if it's an animal, the known contaminated cell line. For disease cell line, we annotate the disease using the NCI thesaurus. We'll probably go and use also the disease ontology once it has all the cancer in it. The site of origin for metastatic cancer cell line, the gene which have been transfected in it using links to AGNC, MGR, RGD, Flybase, and of course UniProt. If a cell line has been discontinued from a catalog, because of course the catalog tells you which cell line you can get, but it doesn't tell you which cell line is no longer distributed by it. And we found this is very important for people to know, yes, this paper speaks about this ATCC cell line, but it's no longer available from ATCC. And if it's registered in an official list, like NIH registry of approved embryonic stem cell. A lot of other things, of course, common misspelling and so on. Some statistics, almost 40,000 cell lines now, from 435 species, but most of them from human, mouse, and rats. And about 30,000 soon synonym, 32,000 reference to almost 8,000 publication, about 80,000 cross-reference to 46 different resources, because there is so many specialized resources, and about 8,000 web links. And all of this is now available, I mean, on either in flat file or OBO format, soon also in OL format, but you can already have it in OBO format. In the ECCB website, which is the oldest running website in life science, because we know that because at the time there was only in 9300 website when ECCB was created, so that's also a nice place to put this because it's where, I mean, SwissProt was put first on the web and so on. And also by FTP, of course, and it's integrated, of course, in our terminology in neXtProt because we use it. Just three, screenshot, you have information when a cell line is contaminated. You see in red, it says problematic cell line. So that the parent cell line itself is contaminated. So this one has not been shown to be contaminated, but if the parent cell is contaminated, then the child line, chances are that it is also, because we also put a hierarchy of a cell line from where it comes from. Of course, as I said, reference, cross-reference, Here you see a line which has a lot of different children, line which were derived from it, websites, cross-reference, and so on. Okay, so I'm almost at the end. I still have 2 slides, and one is to, of course, acknowledge all of the group working at the CALIPHO group. We have first a content part, the biocuration part, which is led by Pascal Godet, which for a long time was also the president and one of the founder. Well, she will always be the founder because for life. She was the president of the Society for Biocuration, for which I make a big also advertisement here, because if you're working on biocuration, please also join societies. There is no contradiction between being member of ISCB and of the, International Society of Biocuration, ISB. They share the 3 letters are in common at least, so you can align them with only one mismatch. So there are a number of annotators. You have a number of software developers, and that group is led by Pierre-André Michel. You have somebody which is very, very precious. All of the people are precious, but we wouldn't be able to do quality work without having Monique, which is basically a whole soul job, and it's a job which is really tough because you're in between the hammer and the nail, is to try to achieve, to make sure that quality is there at every level, software development, in terms of annotation, and so on. And it's, of course, it's everyone's responsibility to do the best job, but she makes sure that people really follows all of the guidelines and quality guidelines that we put. We have a number of people working in the lab, and I'm really privileged to be working in quite synergy with Lydie Lane. So we both are directing the group and working, toward doing those both missions, those 2 missions I described to you, which is to build neXtProt and to also do all of those experiments on protein of unknown function. With this, I will first thank you for your attention and then leave a slide for the end, which is to advertise a meeting, which is the biocuration, annual biocuration meetings, the Society of Biocuration. And this year, next year, sorry, it's going to be in Geneva, and it's also going to be the 30th anniversary of UniProt SwissProt. So, that's going to be, a nice meeting to go. Even if you're not completely into biocuration, please come to Geneva in 2016, and I will be glad to see all of you there. So thank you very much.
Thanks so much, Amos. Are there questions? No? Larry Hunter.
Amos, that's lovely. And there was almost everything you said made my heart soar with joy, but one thing made me hurt. Ooh. And so that was when you described the relations you were going to use in everything. You had this line that said it was homegrown. We had to make it up. I'm surprised Pascal let you get away with that. Why won't you use the relation ontology? What's the issue?
Well, the issue was that, we were very pragmatic. We wanted to use basically something which we could use for all the different projects. And we couldn't find, none of the relation ontology we could find, a different place had what we needed. So we thought we start first with a simple one where we have only 90 terms, and then do what we need, but then try to see if we can work with a relational ontology to try to get the terms that we need and so on. But it was, to be honest, there was a tight schedule to get the protein kinase, work going on, because of course our funders, Marcello wanted this yesterday. And it's the same reason also people ask us, why didn't you use OpenBell, to do it? And at that point, we tried also the OpenBell tools, and we couldn't make them work at the time. It's not criticism of OpenBell. Now it seems that a lot of things work with OpenBell, but we had to really quickly do something. And so we completely willing to scrap completely, it's only 90 terms. So, and we're not wanting to distribute this relationship ontology. We much prefer, to switch to whatever we could get and replace it.
You could use it as the basis of an improvement to the relation ontology. I was ready for you to say it wasn't suitable for your purposes, but it would be nice if we shared, if we could agree on one set of relational terms. Many people need them for many purposes.
I can send you those terms, those 90 terms, so we could see if we could align
Pascal, you take it to the RO. I'm not the right guy.
Thank you. My name is Sirarat, and I am the original creator of the cell line ontology. And I am the lead content developer for EFO right now. So thank you. That was great. So my question would be, is there someone that I can work with in order to transform or help you turn Cellosaurus into an alpha? Because from my experience, it is most useful to sort of link all these attributes together in an ontological forms. So for example, in EFO, we already have a cell line that derives from a cell type from a specific organism. And a lot of those information are reused from the cell line ontology. And we right now have the collaboration that goes on to map EFO to cell line ontology. And also, there are so many types of cell lines. There are permanent primary stem cell lines, and stem cell lines are very hard to define. And there's also another work group with the ontology for biomedical investigation, or OB, that are looking into this as well. So I think would be great compatible partner.
Well, yeah, I mean, so as I said, it's available in OBO, and the idea was to use one of the tools that exists to do an OBO to all conversion. And I was discussing with Chris Mungle on some of the things, and because in fact, OBO format, he criticized it by saying that we were not using, what is it, a way of doing cross-referencing, that we were not using something which I have to admit I didn't know before. And the problem was we're very pragmatic. We have to link to 46 different resources. And a lot of them are not, only 3 are ontologies. So it was easy to do the links and so on. So for the 43 others, they don't even have database like ATCC and so on. So I'm trying to get them already to have standards of using unique identifier and not changing their web links every 2 months and so on. So, I mean, you're welcome to transform the OBO format into all and use it.
What I'm saying is, I can't easily transform OBO into AL. I think it would need a complete turnaround and look into the relationships of things that you have in the Cellosaurus.
No, but relationships, vocabulary is very easy. We only use 2 relationships. It's either a parent, children, or sister line. Everything else, you have to see, this is not an ontology. It's a thesaurus. That's why it's called the Cellosaurus. It basically doesn't want to create an ontology of cell line because I think the people doing the cell line ontology are doing a good work and others. So we don't define, you know, what is a real embryonic stem cell, what it is, where it comes from, and so on. We leave this to the cell line ontology. What we want is a very pragmatic solution. If you're speaking in a paper of this cell line, where does it come from, which species, which collection it comes from, and so on. And then your work, I'm saying not you specifically, all of the people building ontology is to tie up this cell line to the ontology, but then you have a unique identifier for each cell line, and you know that you're speaking about the same cell line, but we're not interested in building an ontology of where this cell line sits in the ontology. You see, it's totally different. The purpose is to have a standardized vocabulary for cell line, which ties up to ontologies, but not to build an ontology. We don't want to be an ontology of cell line.
One last comment.
Yeah?
Yes, so thank you. And I guess then you're trying to build an alpha of this thesaurus?
Sorry?
When you were saying that you already have flat file and OBO format, and now you're working to convert this into alpha.
Yeah.
Okay, thank you.
And we will provide to any format which is useful to people, and so all is, of course, an obvious format we want to do. I mean.
Okay, great presentation. My question is here. Here.
Oh, okay.
Yeah. My question is, how many protein isoforms do you think that's encoded in the human genome?
That's a very good question. And another question which ties up with it is how many of the splice forms are really coding for protein and how many make functional difference? And I mean, the thing is, we know for sure that what is in UniProt KB, because just to explain, what we have in neXtProt in terms of splice isoform is what there is in UniProt. We don't create new sequence data. We don't want to diverge the 2 resources. So we have exactly what there is annotated in UniProt KB as splice variant. And UniProt KB is very now cautious in adding splice variant because they were burnt a lot by the fact that a lot of the splice variant seemed to exist at mRNA level, but then nobody was finding any clues that they do exist at protein level. Now, if you work with some proteomic groups, they tell you the reverse. They suddenly find thousands of new isoforms. So we really are going back and forth between people telling us most of your splice variants don't exist, to people saying you're missing, you know, 3/4 of the splice variants. So I think this is still work in progress to define what is exactly the list of splice variants which are really producing protein, in which tissues express, and how they're making a difference in terms of function.
Yeah, from in this conference, I also accept different information. Some people say, exactly as you mentioned, that some people say there are many, many more protein isoforms. Some people say, well, it's not that big.
Yeah.
So this question, nobody answers.
Well, it's still something to discover.
Yes.
That's nice.
Thanks.
Okay, Amos, thanks again for your lecture. It was wonderful.